Thank you. I'm going to talk about narwhal genetics, peculiarity, and population. And this is a work done in collaboration with Elina Lorentzen at the Globe Institute at the University of Copenhagen, and also in collaboration with many collaborators across the Arctic. Genetics has been a key method in management because with genetic we can define the different population, and then this different population can be considered as the unit which are relevant for management. We can also define the genetic health of a population by looking at levels of inbreeding. But genetics also comes with some challenges because the time scale where we see changes in the genome may be too slow for conservation. And now I'm going to talk about the cases of the narwhal. So here on the map you have the different stocks of narwhal which have been defined under the North Atlantic Marine Mammal Commission. You can see there's, just trying to find the pointer, okay. There's 12 different stocks which are corresponds to the submarine grants. And narwhals migrate between submarine grants, and then they have high fidelity to migration routes to the offshore wintering grounds here. And these stocks have been defined based on different information, including satellite tracking, chemical marker, traditional knowledge, and in some cases genetics. And these different stocks, they show different ecology and behavior, but so far no or little genetic differences have been found. And why is that the case? Here you have a graph which presents the level of genetic diversity of narwhals and other species of marine of cetacean, and you can see that the narwhals here, they have the lowest level of genetic diversity together with the sperm whale. And when there's low diversity, even if the population have separated, it might be difficult to find any differences because there's less material to diverge. But recently, the genetic field has evolved like very, very fast. We went from using a small number of markers and a very small part of the genome to sequence the full genome of several individuals. And this can give us hundreds of thousands of markers across the genome. So with this new method, we have more resolution to test if there are different populations, but also to look at other questions, such as looking at the history of those populations, and we can also look if there's any adaptation to different environments. So here the objective of the study and of the question that I'm going to present in this talk is looking at range-wide population structure of narwhals, and then whether there are different populations east of Greenland. So I apologize for those who've been in the presentation earlier because there's a little bit of overlap in some of the slides. So these are the results on range-wide population structure, so where we've been using whole genomes, and it's only part of the results. So here we have samples from West Greenland, Northwest Greenland and Svalbard, and East Greenland. But we have in total 120 samples with another 100 samples here in West Greenland. But we don't see so far any structure in West Greenland, so I'm going to focus on the data here of the three different populations. And the graph that you have here is a principal component analysis, and each dot here is one individual. And when the dots are close to each other, that means that the individuals are genetically close to each other. And when you have a dot which, like here for example, the orange dot and the blue dot, they are far apart, so that means that the individuals are genetically distinct. So what we can see on this graph is that individuals here from West Greenland, they are genetically distant from East Greenland, and then there's a second level of differentiation when these individuals from East Greenland and from West Greenland are also genetically different from the one in Northwest Greenland and Svalbard. And this is also what we see in the graph here. So the different bars here, it's the different samples that we have, so the different individual narwhals. And the y-axis here is the percentage of assignment of an individual to one of the population. So when you look at the east, the individual sample in the east, you can see they have all 100% assignment to the same population. And we see the same in the west here. All the individual, they have 100% assignment to the west. But when you look at Norwest Greenland, to compare, but these levels are very low. And when you compare the three populations, you can see that the levels of diversity are a little bit higher in Norwest Greenland and Svalbard. And that makes sense because as we have some individuals which have a mixture in their genome from East Greenland and West Greenland. Then I'm going to talk about another study. So this study is the study on the fine scale genetic structure in East Greenland. And I would like to stress that it's preliminary results because it's a relatively low number of samples. So here we have samples, again, like the same sample from Norwest Greenland and Svalbard. We have sample from scorbrisons taken inside scorbrison in the summer, and also sample taken in the spring in Pikialaarfik, and then samples from Pikialaarfik and Pikialaarfik. So what you can see here is the same graph as I've presented on the first slide, so it's the same. So here it shows that the individual samples in scorbrisons in the summer, they are close to each other. Then we have the samples from Pikialaarfik and Pikialaarfik, and then the sample taken in the spring at the entrance of scorbrisons in Pikialaarfik. They are close to the samples that have been taken in Norwest Greenland and Svalbard. I just would like to say that these results are, again, are preliminary, also because the samples from the spring here, they come from a single hunt. And the main results here is that the narwhals which are present inside scorbrisons, which are part of this small and endangered population, they seem to be also genetically isolated. And the Institute has collected, together with hunters, many samples. There are more than 300 samples which are available from East Greenland. So the plan is to sequence more samples from the different locality and season and try to see whether we can see the same pattern, and also hopefully this will help to answer management questions. And then we can also look at the genetic health of the population by looking at level of inbreeding. Hoi